Well, thank you for the opportunity, Yuri, for organizing this and also for the flexibility. I'm not seeing you, so if somebody wants to say something, please speak up. So I will take this opportunity to tell you a little bit about the research going on in my lab. Mainly, we are focusing on immune system aging in human and how this is affecting vaccine responses, and particularly the sex differences in these patterns. So very briefly, so this is a computational immunology meeting, so probably the simple explanation is not needed, but just in case, we have the two arms of the immune system. We have the innate immunity, which is the first and the first line of defense. So the innate immunity is non-specific, so it doesn't, in theory, have a memory associated with a certain antigen, and you have the monocytes, NK cells, dendritic cells in this bucket. And then we have the adaptive immunity, which is slow, but in theory, not in theory, in practice, so it develops a memory. So once an adaptive cell, this could be a B cell or T cell, recognizes an antigen, the next time the cell sees this antigen, it will develop a stronger responses. So the cells that have not yet recognized an antigen are the naive cells, and then eventually they develop into memory cells. So with age, there are some significant changes that happen to the innate and adaptive immune systems. So we see a chronic activation of innate immunity, upregulation of pro-inflammatory molecules as well as cell types, but we also see a decline in adaptive immunity. So this includes loss of naive T cells, which are the T cells that can recognize novel antigens, but also an accumulation of dysfunctional cells, terminally differentiated cells, for example. So together, these changes, both the activation of innate immunity and inactivation of adaptive immunity, reduces our ability to fight against diseases and infections. I have an infection now, so I cannot be there. So in addition, we know that with these changes in the immune system aging, we also lose our ability to respond to vaccination and therapy. So together, these shorten our health and lifespan. So the two questions that I will discuss today: are there any sex differences for biomarkers of human immune system aging? And how do age and sex affect vaccine responses? Of course, the second question is a very big question. I'll just show you one example of one particular vaccine. So let's start with the first one now. So to answer this question, I have been working closely with UConn Health Center on Aging. So our campus is in Farmington, Connecticut. We are not in Bar Harbor, Maine. So we are next to the Aging Center, and through the Aging Center led by George Kushel, we recruited 172 healthy individuals. These are young individuals, middle-aged, and older adults. So these individuals come to the center, they give blood. We get these blood samples from the Aging Center, isolate peripheral blood mononuclear cells, PBMCs, which include the immune cells that I described to you. So it is kind of a blood tissue, if you want, immune tissue. So on these PBMCs, we generate ataxic data for epigenetic profiling, as well as RNA-seq data for gene expression analysis. The project I'll present to you is done using bulk assays, but nowadays, as you also mentioned, we are doing single cell versions of it, which has its own challenges. And we also typically, especially before single cell, we complement this with flow cytometry to understand cell compositional changes and analyze the data. So since here the focus is on sex differences, we paid special attention to balance male and female subjects, and they are matched as much as we could in human studies, both in terms of their age as well as in terms of important clinical factors, frailty, BMI, etc. So in a previous paper, with Jacques Banchoreau and George Kushel, we have uncovered this IL-7R receptor to be among the most affected molecules with aging. So IL-7R is a cell surface receptor expressed on both CD4 and CD8 T cells. So our studies published in 2017 established that not only the IL-7R, but the whole signaling pathway is affected. We also have to specifically CD8 T cells. So here I'm showing you phosphorylated STAT levels upon activation of CD4 and CD8 T cells, and we only see a decline in the CD8 T cells. So this decline in IL-7 signaling is stemming from CD8 T cells. So this is one of the biomarkers we have been using. And when we look at this molecule in this new study with many individuals comparing males and females, first in both sexes, females on the top, males in the bottom, we see a decline in this molecule, both chromatin accessibility level and gene expression level. What was striking was in the case of males, this decline was much more pronounced. And this was not the only molecule. So when we did an unbiased differential analysis using our toxic data, but between old and young individuals, we realized that men are going through twice as many differences compared to women, despite the fact that cohorts are comparable. And this was bulk data, so we did use Chrome HMM states from Roadmap project to deconvolute this aging signature, what is closing and what is opening with age. So as expected, we see a decline in the adaptive cells and an activation in the innate cells. What was most striking between the two sexes was the difference in the level of activation. So if you look at the regions associated with CD4 T monocytes, although in both females and males, these are activated with age, the level of activation is 10-fold higher in males compared to the females. I don't have time, and this is published, so I'm not going to talk too much about that, but we were able to also link it to inflammatory pathways and inflammatory molecules. And in this study that was published just before COVID, we were able to describe what is different, the sex differences in terms of immune system aging. So very briefly, men are going through a more severe immune aging phenotype, where we see a further activation of innate immunity, including increased inflammation, as well as further inactivation of adaptive immunity. IL-7R was one example. And we also see some differences in terms of timing of these changes, which is in alignment with lifespan differences. And we also observed that after 65 years old, the male and female blood immune cells diverge from each other. Older women have more active adaptive immunity, and older men have more active innate immunity. So what are the implications for health and lifespan of the individuals? Of course, the elephant in the room is the difference in life expectancy. In the US, this difference is 5 years. And in addition, we know there are sex differences in many immune diseases. For example, men are more susceptible to infectious diseases and have more severe cases. COVID-19 is one of these examples. On the other hand, women have more likely to have autoimmune diseases. Lupus is an extreme example where 90% of lupus patients are women. And finally, we know there are sex differences in immune responses, although there is a lot more to be described in this last bullet point, and my lab has been studying this for multiple vaccines. And I want to just tell you one example of what we are doing in my lab in this specific part. So how do age and sex affect vaccine responses? So here I will tell you a study that we have started a while ago to understand bacterial pneumonia vaccine responses. So with COVID-19, of course, we don't talk much about bacterial pneumonia, but this is still a very important disease. So 500,000 individuals are hospitalized, thousands of individuals die every year in the US, and most of them are older adults. So there are two vaccines FDA approved and readily adults, Pneumovax and the Prevnar vaccines. I'll tell you a little bit about the differences between them. So both vaccines have a polysaccharide that can bind to the B cell receptor and induce B cell responses. So Pneumovax have this, Prevnar has this. The other thing that Prevnar has, in addition to the polysaccharide, it also has a carrier protein. This protein can be internalized and can be presented to the T cells so that T cells can also respond to this to this bacteria. So that's the major difference. So Pneumovax is just inducing B cell responses. Prevnar is an adjuvant vaccine which can induce both B and T cell responses. Despite significant differences between these two vaccines, we really know very little about who responds to which vaccine, how should we prioritize them, and etc. So to fill this gap, we again worked with George Kushal's Aging Center as well as my immunologist colleague Jacques Banchourou. And we recruited 39 older adults. Everybody is older than 60 years old. And our cohort was split into two groups. So half the individuals got the Prevnar vaccine and the other half got the pneumovax vaccine. And we collected blood from them before vaccination, the day of vaccination, one day after to look at innate responses, at day 10 to look at adaptive responses, day 28 to look at the antibody titers, and day 60 for memory. So this has been a work that Radu and Satya have been working together. Radu is generating the data and Satya is conducting the analysis. So what we have first, before I tell you how we analyze the data, let me briefly explain how we quantify vaccine responses. So very briefly, this is a functional assay. What we do is, this is called an opsonophagocytosis assay. So what we do is we combine the serum from the individuals before and after vaccination with a structure that has phagocytes, which are cells that can eat bacteria. And these cells can eat bacteria better if the bacteria is covered with antibodies. So what we do is we check the system before and after vaccination, and we see the phagocytes' ability to kill bacteria and whether this is improving or not. And this is done for 13 different strains of bacteria. So it's a little bit more complex than flu vaccines, if you are familiar with that. Anyway, long story short, this functional assay gives us pictures like this. So what are we looking at here? So at the top, we see 13 serotypes, different strains of bacteria. And here we are looking at how much increase in our serum's ability of killing we see before and after vaccination. And in the literature, anything above is considered a significant increase, and those are the ones marked in red. So of course, this is nice to look at, but we also need some quantifiable measures to see which individuals are the best responders and worst responders. And we came up with some computational ways to rank the individuals. We look at the number of serotypes to which an individual is eliciting significant responses, which is called the extent, the strength of the responses, not only whether it's threefold increase, but how much increase do we see. And ultimately, we also came up with a way to rank an individual's vaccine responsiveness. So if an individual have a high ranking, like this person on top, this individual is responding to many serotypes with very strong responses. In the bottom, the worst responders have lower ranks and they develop the weakest responses. So this is an important way for us to understand how we can predict vaccine responsiveness, right? Before that, we did first compare, are these vaccines strong vaccines? So here we are looking at Prevnar and pneumovax. And both vaccines, if you look at pre and post titers, both vaccines are inducing stronger responses. And Prevnar is slightly more stronger than pneumovax, but that's kind of expected because it's activating two different cells. So despite this, we still see a lot of individual level heterogeneity in responses, and what could explain that? So of course, one thing is the age. When we look at strength of responses for both vaccines versus the age of individuals, although everybody is older than 60, there is still a 30-year range here. We can see that even within this older adult group, we see effects of the age. So relatively younger, older adults respond better than the really older adults. And the next thing, interesting thing, is the sex differences. So here I'm looking at responses of women and responses of men. So for the Prevnar vaccine, among the top 5 responders, 4 of them were women, and among the worst 5 responders, 4 of them were men. So only for Prevnar vaccine, we observed a sex difference where women are eliciting stronger responses, but no sex differences for pneumovax. So I'll briefly now tell you the concept of baseline predictor. So now using all these measurements and techniques, we know who are the best responders and who are the worst responders, right? So what we can do now is in a way go back in time and look at their baseline immune profiles and to understand what was different in their baseline immune profiles that can help us predict the future, which is their vaccine responses. So we did this for the flow cytometry data. Among the B and CD4 T cell subsets that we have profiled in this study, we noticed that Th1 and Th17 cells are really good predictors of Prevnar responses, but not for pneumovax. And Th1 was positively associated and Th17 was negatively associated, and their ratio was the best predictor. So this is just the same information, but looking at all the subtypes that we studied for CD4 and B cell subsets. So Prevnar responses, Th1 is negatively associated, Th1 is positively associated, and their ratio is the best predictor. So what are these cells? So these are activated CD4 T cells. So when naive CD4 T cells encounter an antigen, in the existence of certain cytokines, it can differentiate into different lineages. So for example, Th1 cells, which is associated with stronger responses, are inducing antibody and cell-mediated responses. On the other hand, Th17, which is associated with weaker responses, are recognized as pro-inflammatory T cells. They secrete pro-inflammatory IL-17, but they also differentiate into this lineage in the existence of pro-inflammatory cytokines like IL-6 and IL-1B. So having high Th17 could be an indication that having high inflammation. So they also co-regulate each other. So Th1 secretes interferon gamma, which suppresses the development of Th17. So because of this, Th1 and Th17 ratio has been also linked to different autoimmune diseases. So obviously these cell types are not completely independent from each other. So then the next question, do we see sex differences in these cell populations? In fact, we do. Women have higher Th1, but they also have lower Th17. And if you look at the ratio, of course, this is much more beneficial for women. They have the higher ratio of Th1 to Th17, which could describe why they are better responding to this vaccine. Similarly, we can use gene expression data from the baseline to predict vaccine responsiveness, and that's what Satya did using WGCNA analysis, which we were surprised. This is probably one of the best correlations I have seen in human studies. But what we have uncovered is there is a cytotoxic gene module that includes all the usual suspects of cytotoxic molecules, NCAM1, granulin, perforin, multiple granzymes. And when you look at the level of expression and the vaccine responses for Prevnar, there is a very strong correlation. So if you have higher cytotoxicity, your responses are going to be weak. And again, this was very specific to Prevnar, the T-dependent vaccine. And when we look at sex differences, this was also in alignment with the responses that we have observed. Women had lower cytotoxicity, which could explain why they developed stronger responses to this vaccine. So to summarize, in this study using systems vaccinology approaches and a human cohort, we were able to describe what predicts Prevnar vaccine responses. So if an individual have high Th1, low Th17, and low cytotoxicity, they will be more responsive to Prevnar. And the other way around will be less responsive. Interestingly, this immune phenotype is correlated both with age and interestingly with sex. Women are more likely to have this phenotype than men. In fact, it's more correlated with sex than age. And where do we go from here? So this can help us consider sex while administering pneumococcal vaccines, but in general, other vaccines too. And ultimately, of course, we would like to move towards precision vaccinology. What vaccine is better for which individual and what are the predictors of that? And these studies can also guide future vaccine development efforts like Th1. There are Th1 promoting adjuvants. Can the next generation of Prevnar vaccines include this? Or can we lower cytotoxicity prior to vaccination to boost the responses of individuals in this phenotype to be able to get better responses. So of course, this is a big team effort. Radu and Satya has done most of the work I presented, but I have been collaborating closely with Jacques as well as George on these studies. And finally, I would like to bring to your attention this meeting I'm organizing in September in Farmington, Connecticut. It's a hybrid meeting. If you like systems immunology, if you want to see more talks about systems immunology, both in aging and diseases, please take a look. And September is the best time to visit New England anyway. So hope to see some of you there. All right. Are you still with me?
Okay. Thank you very much, Duygu, for a great talk. Could you hear the big applause from the audience? And can you hear me now?
I couldn't, but I'm gonna.
Yeah, we did. So one more. Given all the difficulties that
we made it.
Yeah, we made it. Thank you very much. So any questions? We have maybe time for one question or a couple questions. Maybe while you're, and I want to, despite all the challenges we are facing, I really encourage you to do not hesitate and come up and ask questions, because this is a really great opportunity. Even if the speaker is remote, I think they're absolutely ready to answer all your questions. I have a very general, I have a very general maybe question. These are great, very maybe involved studies. What are maybe a couple or one computational challenge that you had to encounter and maybe you successfully resolved it, or maybe you're still seeing that it has to be resolved?
Oh, in the bulk studies, the challenge was deconvoluting the data. And Eladio, who used to be in my lab, established a pipeline where we can use the roadmap states for that, chromHMM states. So that was a good effort. In the single cell, you mentioned the single nuclear attack. That's much more challenging. I didn't get a chance to talk about that, but ASA has developed a very nice package for doublet detection and single nuclear attack-stick data called Amulet, which was published last year at Genome Biology. So I highly encourage you all to take a look if you are using single cell attack data. So single cell attack, we need to establish a lot of pipelines. I'm very comfortable with our doublet detection method, but there are many other things that we need to do, especially in terms of integration with RNA-seq signatures.
Yeah.
So a lot to do there.
Yeah, good point. I agree. Any more questions?